Hello, everybody, and welcome back to my YouTube channel or welcome to my YouTube channel. This is Ruhan speaking, and today we're going to be commencing with the lysogenic cycle. This is part 2 of the video to viruses, viroids, and prions. If you have not watched that video before, you better go over and watch that one first, because if you don't have a clear idea about what the lytic cycle is, you won't know what the lysogenic cycle is about. So, what happens here is that just like in the lytic cycle, you have one loop of DNA. After that, you have your bacteriophage. This bacteriophage has invaded the bacterium and he's releasing his genetic material. Bacteriums only get attacked by double-stranded DNA. Okay. Double-stranded DNA. So this is double-stranded DNA. It's always haywirely coiled. It's not in a perfect shape. So it's not like this. The DNA never looks like this. Okay, it looks like this. So what happens here is that once this viral DNA attacks, there's this kind of a weird thing that happens here. Okay, this is naturally occurring. So what happens here is that you have your bacteriophage. He has released his genetic material. So you have all your material here, and one of the DNAs, they bind to this, to this plasmid. What they do is they bind to this plasmid, okay, like this. So it binds to this plasmid, and what it does to the plasmid is that this is how your regular plasmid looks like, and it makes a recombinant plasmid with it. So this is part of biotechnology. So it makes recombinant DNA in bacteria. So you get your recombinant. Recombinant is basically when you have one complete plasmid and then, it's attached with another one. So when you have this kind of, like a mixture of two plasmids fixated into one plasmid, it's called a recombinant DNA. So you get a bacteria with recombinant DNA now. So what happens to this bacteria is, because the plasmid is, it's recombinant now, but this is how its genetic makeup looks like. So this is how its genetic makeup looks like. The bacteriophage is still here. So now that the lysogenic cycle has commenced, what happens to the bacteria is that he divides by binary fission, because bacteriums divide by binary fission. There are a total of 5 ways in which a bacterium can, reproduce. Binary fission is the most common one. So you get all your daughter cells which are formed, all of them, they have your recombinant DNA. These ones divide into 2. This one divides into 2. Then this one into 2 and this one into 2. This one into 2 and this one into 2. So this is how the cycle, this is how the cycle continues. So this is known as your cycle. This is your binary fission cycle. This is how it looks like. The circle which I'm putting around 2 strands is to show that both of them have this process going on. They'll keep dividing into 2. So this is how the cycle takes place. And then you have a lot of bacteriums, and all of them are covered with the recombinant DNA. Okay. All of them have this recombinant DNA in them. So that was the lysogenic cycle. What we have next is viroids. What exactly is a viroid? So a viroid is a single strand of RNA. Okay, it's single stranded RNA, ssRNA, that is devoid of its capsid. So let's go over the anatomy of a virus once. So you have your virus here, and then you have the DNA inside. Now this is single stranded RNA. So this is how it looks like. This part and this part. There are only 2 parts which you have to note here. This part is called your this is your capsid and this is your ssRNA. So after all of this happens, what happens to the viroid is that this capsid goes away. Okay, it's only single-stranded RNA. So you have one piece of single-stranded RNA fallen down. It has very low molecular weight. This is one feature. We can call this a feature or a property. A property. So this is the property of the viroid. Then T.O. Diner. So T.O. Diner is the person who discovered the first ever viroid. And he discovered it in potatoes. So what happened is he found an abnormally shaped, like it was a spindle shaped potato. It was, how does a potato look like? This is how a normal potato looks like. It has buds some places, but this is how it's like an irregular sphere. But then what he found was spindle shaped potatoes. So however a spindle looks like, this is how the potatoes looked like. So he got confused that if this is the way a potato looks, then these aren't potatoes, there's something else. But then they were indeed potatoes. So he got confused about what exactly is happening here. So what he did was he conducted a lot of research on it, on the potato. He does everyone remember discovery of the virus like we did? Who was the person? What was his name? His name was DJ Ivanovsky. Okay. This is present in my last video. You can go and watch it quickly. It's about DJ Ivanovsky and how he found out the first ever virus. And it was the tobacco mosaic virus. So what he did was he took plant sap and then he passed it through bacteria proof filters, and that's all he did. And then he kept searching of what was the organism or what was the toxin that was causing this issue. And what he concluded was, what his partner M. Vijnik concluded was that this is infectious living fluid. It's contagium vivium fluidum. That means it's a infectious living fluid. It's not a living thing. It's an infectious fluid. But then W. Stanley, another scientist, what he did was he crystallized the sap, and what he found out was that there was some kind of powder left behind, and that was your virus, because viruses have a property of being allowed to have crystallization take place. So he crystallized the virus and he found out that it consists mostly of proteins, which is your capsid layer. Okay. I said DNA, RNAs, ribonucleic acid. So this is another thing, but capsid is completely made up of proteins. It's a polymer only. It's a naturally occurring polymer. So after this, going back to the future, what happened here is that T.O. Diner found out that he got spindle shaped potatoes. He did a lot of research and he found out that the virus, it wasn't crystallizing. So what exactly is that? That means there's no protein, there's very little protein present. So it's a single stranded RNA, which was causing the issue in the potato. Okay. So a normal potato was germinating into a spindle shaped potato. And that was the reason why. This is how your viroid was found in, it was found in spindle shaped potatoes, viroids. So viroid is nothing but a single strand of RNA, infectious RNA to be, exact. Now let's go over to prions. So what is a prion? Viroid, prion, they sound the same and they are the same thing. They're a little different. So prions I'll be writing with pink. So what happens here is that you have your normal viral DNA and you have something like this. Okay. The genetic material here doesn't matter. It can be single-stranded DNA, single-stranded RNA, double-stranded DNA. So this capsid layer which is there, that stays, but your RNA or whatever genetic material is there, it goes away. So you only have your leftover capsid layer. So that's only protein. Okay, so it's an abnormally shaped protein that causes diseases in other organisms. So this is how a normally shaped protein inside a healthy person looks like. Inside a health. So right now we'll be analyzing the mad cow disease, bovine spongiform encephalopathy. So the simple name for it is mad cow disease. So what happens here is that this prion thing is there. So it's, it's a very small thing. It's not visible. So it lies around everywhere. It's an abnormally shaped protein. So what happens here is that your normal protein is shaped in a, it has a regular shape. It's following some sort of pattern. All polymers follow some sort of pattern. They have repeating units. So this one is properly shaped, and this is a body protein. It's somewhere in the body. But here, what does MAD denote? Something related to brain, something to do with neurology. And encephalopathy is also something that relates to brains. So what happens here is that this prion which is there, it enters the cow's body. Okay, it enters the body. It triggers the body to create abnormal proteins in the body too, rather than the normal shaped proteins. Okay, so what happens is you have your normal shaped proteins, the ribosomes and other protein creating, organelles are creating proteins. But then this prion, what this does is it triggers this ribosome to create abnormally shaped proteins. So instead of this normal one, the protein you get looks something like this. And then, our body is protein based. We need protein to create cells, to create new cells. Protein is what? And nitrogen is needed to create proteins. So you, in your brain, when these proteins are being created in the cow, what happens here is, so to the cow, what happens is her brain, which is there, this is how a normally shaped brain looks like. Her brain will start deteriorating because it's an abnormally shaped protein. So it's causing, it's causing chemical imbalances in the brain, and it turns the cow mad per se. It turns it mad in double quotes. So it turns her mad. So what happens later is, so to break down BSE, this is your scientific name. Mad cow disease is a very layman's term. Bovine means cow. Spongiform means spongy brain. So the brain turns very spongy. Brain has consistency of butter, okay, of, room temperature butter, which is pure butter, but it turns into a spongy thing. And encephalopathy has something to do with neurology. So this BSE, so people, I mean, in India, this is a very rare case, but there's other countries who have this shock of eating beef. So what happens is, so when a person eats beef, when he ingests beef, what happens is that this prion is present in there. Okay, it spreads around the body. The whole body starts making, because brain is, it's the coordination center. So it triggers all other protein creating organelles to start making prions. And this goes into your beef. And then what happens is this protein goes into human, and it causes CR Jacob disease, which is the human name for a mad cow disease. Okay. Most prionic diseases are neurological. Now, this is a fact. This is a proper scientific fact that most prionic diseases are neurological. And we have approached the end of our class. So, end of this video rather than a class. So this was a very short video. We just went through the lysogenic cycle, through viroids and through prions. I hope you enjoyed the video. Please give it a like and hit subscribe. Thank you so much for watching my video. Goodbye. See you in the next one. So the next video, which I'm planning to make a short session on my further videos. So I'll release this video on Instagram too. What I'm deciding to make a video on next is the plant kingdom, which there's a lot of stuff to cover there. You have bryophytes, pteridophytes, gymnosperm, angiosperm, and algae. So what I'm thinking of is making one video on algae, one video on bryophytes and pteridophytes, and one video on gymnosperms, because angiosperms is not that necessary because there's flower producing plants. But then that's my idea. I'm going through the animal kingdom right now, so let's not expect a video soon for the animal kingdom. Yeah, that's it. Thank you so much.